స్టార్ట్ చేసేయచ్చా? వచ్చేసారా స్పీకర్?
కాల్ చేసా మేడం, ఆయనకి ఆల్రెడీ వచ్చేసాను అన్నారు.
స్లైడా ఇది? దేబ్నాథా? సురజిత్ కాదు, దేబ్నాథ్ అంతేనా? ఇక్కడ వీడియో ఆన్ అవ్వట్లే లేదా? వీడియో
Okay, so shall we start or shall we wait?
Sir, just a minute, sir.
Right, okay.
Surajit sir, good morning. Surajit sir, can you hear me?
Yeah, good morning.
Sir, good morning.
How are you, sir?
How are you, sir? Fine, sir. Thank you.
Yeah, good morning.
Yours is the first
Okay.
session, sir, today.
Okay.
Amina Gadu,
Sir.
you start.
Thank you, sir. Okay, sir. Good morning, all. I welcome you all to the 4th day of ISTE ISTE sponsored role of artificial intelligence in drug discovery and development. Myself, Dr. S.K. Aminabi, Associate Professor, Department of Pharmacology in VV Institute of Pharmaceutical Sciences. And today I am the co-chairperson for this session. I request Principal Sir and Coordinator of this ISTE ISTE sponsored role of artificial intelligence in drug discovery and development to speak few words. To you, sir.
Okay. Thank you, Amina Gadu.
Thank you, sir.
A pleasant good morning to all the participants for the day 4 of 1 week online AICTE ISTE sponsored refresher program on the role of artificial intelligence in drug discovery and development, organized by VV Institute of Pharmaceutical Sciences, Gudlavalledu, Krishna District, Andhra Pradesh. And today we are having a 3 plenary lectures. First session is takeover by Dr. Surajit Debnath, Head Department of Medical Laboratory Technology, Women's Polytechnic Tripura, and he will deliver a talk on hands-on training on applications of artificial intelligence and machine learning for biomedical research. He is having a vast experience in various webinars and seminars and conferences in relation to the artificial intelligence. Okay, that's why it is a different session planned with the support of resource person as a hands-on training for the benefit of the participants. Okay, the chairperson for that particular session is Dr. Sheikh Aminabi, Associate Professor. And for session 2 is takeover by Dr. A. Sridevi Garu, Professor, Institute of Pharmaceutical Technology, Sri Padmavati Mahila Viswavidyalayam, Tirupati, and she will speak on computational screening and evidence-based support for in vitro antioxidant activity of Jasminium auriculatum. And for the chairperson for this session is Ms. B. Satyasri Garu, Associate Professor. Next, the session 3 is taken over by Dr. P. Ramalingam Garu, Associate Professor and Head in Charge, Department of Pharmaceutical Analysis, National Institute of Pharmaceutical Education Research, Naipar, Hazipur, Bihar. And he will deliver a talk on computational approach in the prediction of analytical behavior of analytes. And the chairperson for session 3 is Mr. T. Prashanti Garu, Associate Professor. And now I hand over the session to Dr. Sheikh Amina Garu, Chairperson for the today's session 1 and Head Department of Pharmacology. And she will introduce the resource person to the participants. Over to Amina Garu.
Thank you so much, sir, for explaining in a detailed manner the date schedule of today's day. I take it my pleasure to introduce today's speaker, Dr. Surajit Debnath, sir. Dr. Debnath, sir, currently is the head Department of Medical Laboratory Technology, Women's Polytechnic, Tirupura, with more than 13 years of experience in biomedical research and high-level teaching. Sir participated in various academic works in institutions of Germany, Denmark, and Hong Kong, besides in premier Indian labs such as AIMS, IIT Kharagpur, IIT Guwahati, and NBPGR. Sir is a recipient of Young Scientist Award from different agencies like DBT, DST, and CSIR. Recipient of Tata Trust Grant for CME course in Royal College of Pathologists, London. Reviewer of several Scoopers SCI indexed journals published by Elsewhere, Springer, Biomed Central. Along with regular academic activities, he is also engaged with several biotechnology companies in northern and western India as a consultant. He, was, he have conducted various workshops and talks, acted as session chair in international conferences sponsored by national and international academic agencies. He delivered numerous invited talks in national and international academic gatherings. Currently, he is guiding a group of international students for their internship from Russia, Turkey, and West African countries. Dr. Debnath filed 2 patents in 2021 in India on biotechnology. With this brief introduction, now I request Dr. Debnath sir to take over the session. To you, sir.
Yes. Thank you, madam.
Thank you,
Yes.
sir.
Thank you, madam. Thank you, sir, for giving me the opportunity to discuss in this August gathering. So I shall be sharing my PPT and eventually we'll start our this discussion. So just madam, let me know if things are visible and audible and everything.
Yes, sir. Yes, sir.
Uh,
I'll let you know, sir.
okay, right. So okay. Okay, madam, just let me know whether things are visible.
Yeah. Your screen is visible, sir.
Okay. And I'm quite audible, right, madam?
Yeah. Your voice is very clear, sir.
Okay. Okay, absolutely fine. Thank you.
Yes,
So
yes.
today, in the morning session, these are very challenging because participants will feel a little dizzy, and things will be like in order to make more effort should be given so that participants will be engaged to the session thoroughly. So the discussion today will be we're talking on applications of artificial and intelligence and machine learning for biomedical research, and I'll have divided the whole session in two parts. First of all, although the whole FDP is based on drug discovery, but we must not also forget that there are other aspects related to it. So once we talk about drug discovery, we are not essentially talking only about the small molecules and chemotherapeutic drugs. We are also talking about drugs that are peptides in nature. For example, vaccines. Before that, one more important aspect. I am sure all the participants will definitely agree with that. That the diagnostic part is also a very important aspect in biomedical research. So eventually, what has happened? This particular domain of computational intelligence or computational learning process has been applied in these fields in a significant way. Now, what is the rationale? Why we are everybody is talking about artificial learning instead of human learning? Because of the simple reason that machine learning is much more cost effective, or shall I say cheap, compared to human learning. So what will happen if we can train a certain amount of algorithms to do the repetitive task with high precision? So what will happen? Many a times it has been seen that human intelligence have some limit while doing a repetitive tasks. So in that case, if we can automate the whole process, what will happen is eventually the overall application becomes highly systematic as well as cost effective. The cost of putting a trained person on a particular platform is much more than that of the machine intelligence, deploying tools of machine intelligence. So today's discussion will focus on how exactly this artificial intelligence and machine learning and some of these aspects can be applied for cancer diagnostics, therapy, as well as development of vaccines. Okay, so I will do a little hands-on, although a full-scale hands-on may not be possible, but I'll share with you some audio-visual descriptions where you will able to find out that which kind of tools are useful and how these tools are useful for these two aspects. Okay, so with these, I shall move on to the next slide. So it has been seen significantly that in recent times, the number of intellectual property that has been generated through the application of machine learning and artificial intelligence has increased to a significant extent. Why is this so? Because everybody understood that to automate certain processes, if we can deploy machine learning tools and artificial intelligence, it will be much more beneficial, and eventually the benefit of the whole process will be given to the patient. So patient party need not to invest a lot many more funds towards the diagnostics and therapeutics. If we can deploy some of the machine learning intelligence tools and algorithms for doing the initial stages where your objective is to minimize false positives as well as false negatives. So we'll come to those points in a detailed way, and there has been excellent boom in this particular domain. This is a very simple example. So in prediction, what exactly happens? In prediction, for example, this is a simple machine learning algorithm where the algorithm tries to find out which is a malignant tumor and which is not. Okay, which is benign or localized, which can be get rid of by a simple procedure or some kind of surgical treatment, but it won't be life threatening or fatal. So how do the machine learning algorithm will do it? What it will do is it will classify the size of the tumor. So it is just an example. It could be different other aspects as well. So in this particular example, what the machine learning tool, what it is doing is it is it is classifying two different kinds of cancers on the basis of the size of the tumor. If the size of the tumor reaches a certain level, so it is classifying it as a malignant tumor, and if it is not, then it is classifying it as a benign tumor. Now the problem here is the false positives and false negatives. As you can see here, if in the this thing if the answer is malignant, yes, and that case also there are several what you call is false negatives. Whereas if the answer is no, there are several false positives. So the overall objective should be, you cannot make a fuzzy assumption, because these are life threatening conditions, and nobody should have some fuzzy assumption. It should be completely clear. It should be either positive or it should be either negative. So there are certain drawbacks of machine learning tools and techniques. So it is the responsibility ability of the researcher to make the algorithm so good that the number of false positives are significantly reduced. So on this particular domain only, our small lab is working towards this. Although there are significant limitations in our side, but despite of that, we are trying to put as much effort that can be possibly done to answer these kind of questions. So one more thing that is very important these days is the generation of data. So it has been seen that recently, if you can see this, there has been rapid increase in the amount of data that is being generated. So it could be multiple different types of data, including biomedical data, genomics data, proteomics data, which is huge in number. So in order to handle all this data and to find out informations that are really needed out of this huge pile of data, good data science tools and machine learning tools are needed. So this is true for biomedical data quite significant way, because every day, for example, in our lab, although we are a very rudimentary lab compared to the national facilities that are there everywhere, in Northeast we are having so much of stringency anyway. So every year we generate a lot of biomedical data. We are having specific collaborations with hospitals, and therefore a number of data in terms of bioelectrical signals, EEG, ECG, then multiple different MRIs, CT scan, and a lot many more we generate and we preserve. So we also think that there should be some tools and techniques which will help us to accumulate the exact information that we anticipate to know in order to classify certain image files or to identify certain diseases and to do a lot many more work that can be done using these machine learning tools. So this is again is why this particular theme is necessary, and I congratulate our this thing organizers to put a specific terminology where they are applying machine learning and artificial intelligence for drug discovery. So here again, so where are the domains? What are the domains that are generating more and more numbers of data? So one of that is biomedical data. Okay, high throughput sequencing. Now, high throughput sequencing, we are talking about genomics as well as proteomics, because we are also talking about X-ray crystallography of proteins. For example, let me give you a very simple example of the COVID-19 proteome, the whole proteome I'm talking about, the spike proteins, the internal proteins, and everything else. So there is a huge amount of data that is being generated every day, and therefore very good data science tools are desperately needed. So till now, in the conventional way, how exactly this cancer diagnosis takes place? So cancer diagnosis usually takes place in a way where we do a FNSE, which is fine needle aspiration cytology, in order to accumulate certain amount of tissue from the cancerous tissue, and then we do a what do you call is microscopic analysis to find out. We call it histopathology, in order to find out where exactly the tissue has gone wrong. Is there any abnormality in terms of numbers of chromosomes? That is there any aneuploidy, polyploidy, or things like that? And then eventually we look for, is there some certain kind of what do you call metastasis that has taken place, in order to find out what is the stage of the cancer. So all these things are usually done manually, and which require a lot of skill. So let me tell you, for to train a proper histopathologist, it takes 5 years of basic MBBS and then specialization in MD. So overall, the whole process becomes very, very tedious, and therefore the cost of the analysis is also very high. Okay, so is there a possibility that we can automate the whole process? So let us see a video where we will show you. Most of us already knows about that, because most of us are belong to this particular domain of biomedical thing, allied thing. It could be either pharmacy or something else, but most of us already knows about it. Still, I shall request our coordinator, madam, to
Sir,
please play the first video, so that we will have a interacting thing, so that we can understand how exactly things goes, and what we can do by applying machine learning applications. Madam, the first video in a mute condition. मैडम थोड़ा सा पहले से थोड़ा दीजिएगा, a little bit behind. Madam, would you please, yeah, a little bit this thing I missed. A little bit early, madam.
Sure, sir. So can you tell me from
Yes,
this onwards?
from beginning only, madam, no problem. So this video has been produced by a third party, but these are freely available, and therefore we can definitely talk about it. So this is the tissue where the cancer has been developed, and therefore from that tissue we have isolated certain material and made into a slide, and then put it under microscope. And before that, we need to stain the thing in order to increase the what we call is contrast, and this is how it looks like. Okay, now this is nothing but the histopathology of the thing. Now how this thing has developed internally, the genes are there, which is basically the main regulator of this particular cancer. So what exactly is done is, we cut the tissue in small pieces and then observe under the microscope. But while doing so, we do not consider about the genes that are present in this. So this is today's approach, where these are the DNA material that is they are in the genome, and which are made up of specific nucleotides, which contains the information that why exactly and which type of cancer there has been developed. Okay, so initially we are not looking at the genomics part, and we are only looking at the phenotypic condition of the tissue where the cancer has been developed, but we are not talking about the genomics part. But we know that anything whenever a cancer develops, it is due to an abnormality in the proto-oncogenes, that is at the genomic level, and it only determines that which kind of cancer it will develop. So that was the conventional method. But now we shall enter into a phase where the genomics things will also be considered. So, madam, thank you for your this thing. You can unshare now. So, what exactly happens is, in today's case, we shall have an opportunity to discuss the genomic part as well. So, what will happen? We are not denying that there is no use of the histopathology, which is very tedious to perform. But what we are trying to say is there can be very good what do you call patient compliance if we go along with that the exterior image processing and the histopathology. We can also consider the genomics part. So how exactly happens these days? These days, one more approach has been there by using machine learning, where they are talking about the image processing. In image processing, what it does is the repetitive task of identifying a particular area where cancer has developed, we are putting it to the computational algorithm. So how exactly we do this? There are three types of images that are usually taken. One is structural imaging, where we are taking radiology, MRI scan, this and that. Then eventually there is one more functional imaging, where some MRI and other things are there. And finally, the optical imaging, where we are doing the histopathology or the tissue sectioning and observing under microscope. Now, all these things generates a lot of image data. So this image data can be classified by means of an image processing tool, which is nothing but a machine learning tool itself. So in machine learning, what exactly happens? You put some instruction to the learning algorithm so that it can classify all by itself. So we'll come to those details, what exactly happens. Now, in that case, there are some limitations, as I have given you earlier. What is the limitation? It can predict wrongly. Okay, because some minute details may not be incorporated in the intelligent pathway or the algorithm, and therefore it can be get missed. But we cannot take any chances. So, what is our objective? Our objective is to reduce all kinds of false positives as well as false negatives. So while doing so, it has been seen that all kinds of artificial intelligence tools, or shall I say algorithms, including naive Bayes algorithm, then genetic algorithm, fuzzy logics, then clustering methods, then decision trees, and then support vector machines and artificial neural networks. Now, these are the couple of tools which has been used with a significant application by machine learning and computational science experts in order to predict and classify cancers. Now, out of that, it has been seen that artificial neural network has exceptional ability to classify and predict the outcome of a cancer in a very good way, okay, with much more precision. So out of all these 6-7 algorithms, artificial neural network works best. So it has been reported by another group of scientists, but we are taking it as our this thing benchmark. So using that, one more lab has developed a very good strategy. So what is the strategy? It will give us some idea how exactly this machine learning things works. So in that case, there was a particular case of brain tumor where the tissue section has been made into a slide, as you can see here, and then eventually put it under the microscope. Now, from that microscope, the whole algorithm is such that it will identify certain areas of interest, okay, and then try to collect as many as data on the areas of interest, and then put it into a convolutional neural network. Now, in convolutional neural network, what exactly it can do is it can predict how long the patient will survive if the patient is having this kind of structure in his brain tube. Are you getting? So I'm talking about cellular level structure. That is histopathology or FNAC or biopsy, usually we call it. So on the basis of the image that has been generated through the biopsy, the artificial neural network pathway can identify or can predict how long the patient will survive. Now, this report has been published in PLOS One recently, 2020, and it is one of the best benchmark of using artificial intelligence for cancer diagnostics. But it is not only that. I'll come to the next point. Why this is important, this particular example. So let us talk about a little bit about the artificial neural network, how exactly it works. So what it does is there are several hidden layers and there are some input layers. These input layers are nothing but the image that you are feeding into the system. Eventually, it will try to predict what exactly it means, what exactly the figure means. So by doing this, it can try to identify as well as predict the stage of the cancer and then do a back propagation to find out how long the patient will survive. This is an excellent case where machine learning has been used for biomedical research. In a more systematic and more what do you call understandable way, we can put it like this. Now, this example is also very good if you can see it in a proper way. So what exactly it does is, what is our training set? Now, training set is nothing but you are making the algorithm to learn how to identify the digit 8. Okay, so in order to do so, what you are doing, you are trying to make the algorithm to learn all these images which looks similarly like the digit 8. So what it will do is, anything which is similar to digit 8, it will predict as digit 8. Okay, so this is how the nodes and the internal what do you call technology of artificial neural network works. Okay, so on the basis of this, image processing can be done in a very good way, and by looking at a particular image, it can predict what is the stage of the cancer. Okay, but in order to reduce the false positive, as I have said earlier, so here also you can see, I'll give you a little this thing to understand our participants. So as you can know, that digit 8 and the digit 0, if we write in this way, will be much more similarity, isn't it? So what will happen, this particular neural network which you have designed to identify digit 8, it can wrongly identify digit 0 as digit 8. If the information is not there in the artificial neural network, that digit 0 also looks like digit 8, and there are some superficial differences. Understood? So in that case, it will be a risky proposition. So in order to avoid that, what exactly we can do? Now see here, what these people have done. They have not only identified the images on the basis of the histopathology or the biopsy, they have also incorporated genomics into it. So this part is very, very important, where our lab is working towards. Anyway, so before going to the genomics part, let me tell you that there are some benefits as well as limitations of artificial neural network. This is not inconceivable, okay, invincible. So if our benefits outperforms the limitations, then only we'll use it. And it has been demonstrated in multiple times, multiple incidences, that artificial neural network will be the best available tool which will give you the next generation ability to predict. Okay, so now we are calling it deep learning. I am very much sure most of you have this thing. In deep learning, what exactly happens? You just a very simple what you call input. The input is also very, very limited, and the output is exceptionally correct and precise. So deep learning, the roots of deep learning is on artificial neural network only, out of all those 7 algorithms we have shown you. Now, what this particular lab has done, this is very interesting to see. So what they have done, they have given a bar chart, if you can see it. So in the bar chart, you can see 4 different what you call results. Out of that, one particular result is the red one, if you can see. So what this particular result is, it is using artificial neural network and image processing along with the genomics. So what exactly has happened, I'm telling you, there is a gene called IDH. Now IDH gene is responsible for the growth of the cells in the brain. So if something is going wrong, if there is a mutation in IDH gene, it will eventually lead to certain kinds of brain tumor. Okay, there are different classes of this. I'm not going into that. A particular kind of brain tumor. So what this particular group has seen, that if we add image processing of biopsy images along with the genomics, or that is sequencing the gene and identifying whether mutation is there or not, it will have an excellent predictive what you call capacity, and it can pinpoint exact days, how many days a particular patient can survive. And this is a wonderful achievement of artificial intelligence in biomedical research. So I could not this thing resist the idea to talk about this particular this thing what you call achievement of this particular fraternity in this particular session with you. So in that particular result, what it is showing? It is showing that if I use only artificial neural network on image processing, our results are quite low. The first bar chart, you can see, the result is not very good. But when we incorporate both, that is the genomics as well as artificial neural network, the significant achievement or the prediction is so precise that it can exactly identify how many days a patient will survive on the basis of 2 things, on the basis of the genomics, on the basis of the biopsy image processing. Understood? So this is a very good example. In 2020 only it has been published, and it has been talked about everywhere. So what is the use of incorporating these things? Why we are using artificial neural network and artificial machine intelligence and so on? As I have given you, please understand the main role of artificial intelligence and machine learning in drug discovery, drug design, and then predictive analysis, diagnostic, and all these things are to reduce the cost, so that the cost is not borne by the patient party, so that patient party gets some relief, and our prediction also becomes very superior. So that was the main idea. Now while doing so, we are also talking about something which is precision medicine. So what exactly is precision medicine? Precision medicine is delivering a therapy which is exactly needed and which is customized for a particular treatment. It is not generalized. So a simple kind of customization or precision is also like this. For example, an adult individual is suffering from some symptoms of COVID, that is fever, body ache, and this and that. So what will happen? This person will be given a paracetamol of 650 mg per this thing per day, isn't it? But if the patient is like the body weight of the patient is less than 50 kilos, he will not be given 650 mg of this thing, what do you call paracetamol. He will be rather given a 500 mg, isn't it? If the patient is pediatric, then a different kind of this thing is given of the same medicine. Only the amount of dosage is different, isn't it? Or the form of the dosage, how you are delivering it, whether you are putting it suspension for a pediatric child, or you are giving a sustained release tablet. So this is also a kind of customization, or shall we say precision. But we need more precision than that. what exactly the precision will be? For deploying this kind of precision medicine, we need to invest more on artificial intelligence. So this is a classical demonstration of how precision medicine works. In the first case, if you can see, the same medicine is given to three bunches of people. So one group of people, they are showing very good result. In some people, there is no result at all. As you can see, the x and y axis, there is a straight line. But in some patients, it is decreasing the efficacy of the drug, or there is no response at all, or there are some contraindications. With paracetamol also it is true. For example, some people are there who are hypersensitive to paracetamol. So what exactly happens is they initiate a process which is called drug mediated hemolysis, drug induced hemolytic anemia or something like that. So in those patients, what happens, whenever they'll take paracetamol, the rbc number will decrease suddenly, and the patient will undergo a shock, isn't it? So those things are there. But in precision medicine, what exactly happens is each and every individual will be given a particular kind of therapy so that we can reach an optimum or more than optimum result, and everybody will get what do you call a relief from the disease symptom. So that was the idea of precision medicine. So in order to do precision medicine also, we have to deploy machine learning and artificial intelligence tools in a quite big way. So we'll see to this also, that how exactly this precision medicine will work. I'll once again request our coordinator madam to please play the second video, so that in a quick look we can have on how exactly precision medicine will work.
Sure,
So
sir.
the objective of this sharing with you the audiovisual thing is only that it becomes more interactive and you find it interesting, because in the early sessions of the morning, madam, please share the second video in the mute condition, so you can see that what exactly happens when we are talking about precision medicine, how good it is in cancer diagnostic and several other things. Okay. No.
Wait, sir.
Okay, skip it, madam. So this video has been produced by University of Monash, Australia. In an excellent way, they'll show you how exactly happens in this. We are not talking about this particular strategy, but we are talking about the concept behind it. Now, this is a fluorescent microscope. Now, fluorescent microscope has a very good ability to differentiate good and bad cells, as you all know, on the basis of the fluorescent signal it receives. So now here you see there are two kinds of cells you will be seeing. The fluorescent has been switched on, that is the UV ray has been switched on in the fluorescent microscope, and two types of cells you can see. One, the nucleus is pink, and another, the nucleus is blue. Now this blue nucleus indicates that there is something wrong going in the nucleus. What is that? It could be either polyploidy, it could be eventual what you call entering of one certain cell into another, this kind of thing. In those cases, what we can do is we can deliver a medicine quite precise way, so that it will be delivered to this particular cell only by a process called receptor mediated endocytosis. Okay, so what exactly happens is this particular drug will be targeted to this particular cell only by a concept called drug targeting, so that it will be acting on this particular cell but not on the other cells which are on the periphery. Understood? Now this is the drug that has been delivered into the cell, and now it is entering into the exact target site. Okay, as you can see, the endoplasmic reticulum, Golgi body, everything is bypassing, and it is entering into the nucleus. So this large ball is the nucleus, and now it has entered into the nucleus. Okay. So what exactly happened is the targeting is so good that it is entering only in the nucleus of a certain kind of cell, which has given a abnormal fluorescent signal. Okay. So now the drug has been released. Okay. So till now see how good the sustained release concept is working here. So eventually what will happen? The cell will die due to a thing called programmed cell death or apoptosis. So what it is doing is, okay, thank you, madam. So what it is doing is, it is doing a thing called, uh, okay, just 1 second, madam. Okay, thank you, madam. So what exactly is happening here is we are targeting the particular cancerous cell in so good way that there is no interference, there is no what you call abnormal drug targeting. Otherwise, what exactly happens? Most of you already know. For example, how exactly shall we say aminopterin works? That is a folic acid analog. So folic acid is a very important raw material for production of DNA by de novo DNA synthetic machinery. That is a purine pyrimidine pathway. So if we do not give folic acid, so what will happen? No more DNA will be formed. So in chemotherapy today, what is the drug treatment? What is the treatment of cancer? The treatment of cancer is chemotherapy. Most of the chemotherapy is nothing but folic acid analog. For example, aminopterin and many more. So what exactly it does? The patient will be taking an injection, and so the folic acid analog will enter into good cell, bad cell, every kind of cell, and therefore there is a significant what we call contraindication of multiple things. For example, there will be heavy nephrotoxicity, hepatotoxicity, and then rest of the multiple different things. Why? Because the targeting is not good. The drug is not reaching the cancerous cell. It is also reaching somewhere else where it is not necessary. So, what is the role of today's precision medicine? By applying machine learning tool, what we can do is we can predict how exactly and where exactly we have to deliver and which kind of vehicle we should use. Should we use a liposome mediated delivery? Should we use a intravenous or intradermal injection? Will it be a oral medication with sustained release concept and things like that? Everything can be predicted so that yeah, I'm sure you have learned about QSAR and then Lipinski rules and things like that. We are not going to those details. So what exactly we are trying to understand is how well precision medicine can work if we can automate certain repetitive process in order to predict certain technical bottlenecks that we are having today. We cannot use a very good precision targeting, and some examples are only there. For example, monoclonal antibodies and things like that. However, these are not standardized for all kinds of cancer. So coming back to the theory, so how exactly we'll do that? So in order to do that, we have to have very good efficiency in what we call genomic sequence analysis, or shall we say gene sequencing. So today's gene sequencing are done by multiple different methods. I'm not going into the detail. It is a simple representation that how whole genome sequencing and then this PCR based sequencing are taking place, and shotgun sequencing are taking place. So what they are doing is they are identifying a particular kind of mutation in a very highly precise way. A single nucleotide polymorphism can also be identified. So these will help us to understand why exactly precision medicine is needed. Now, whenever we are talking about genomics, we should also talk about the database that is putting all kinds of genomic data. So in this case, what exactly happens is all the reference genomes are present in this particular database, which is free for everybody. So this is also a development where computational science has given us excellent opportunity to not only generate data but also to preserve it as a soft copy, and it can be retrieved in a real time basis, and it can also update it at a real time basis. So every day this particular database is getting more and more number of data where we can use our tools to predict the result of a particular SNP or single nucleotide polymorphism. Okay, so we'll come to this thing also. And this is the Human Genome Project. Now, in Human Genome Project, as you all know, it started in 1990s and then eventually completed in 10, 15 years. And then what has happened? All the reference genomes data we are having. Okay, so out of these all genomes, these are having the information for everything: the skin color, height of the person, how much hemoglobin he will have, what will be the color of the hair, and each and every detail. Now, including all those exterior details, some interior details are also there. So let me give you an example of this. Now, this is a detail of a particular gene which is called BRCA. So BRCA is a gene which is responsible for production of an antigen on the surface of the breast cancer tumor. So what exactly happens is BRCA gene is a proto-oncogene which is responsible for growth and development of human breast or human mammary gland in a certain way. So if something goes wrong in this particular gene, what will happen? It will lead to a breast cancer. Okay. So what is the procedure of detecting breast? We shall come to that point also. So what exactly happens is if we can identify any mutation in BRCA, it is directly related to, or it is having high probability that the patient will have a breast cancer. Okay, so most of us already knows about a very celebrated person, Angelina Jolie, who was a Hollywood actor. So she had a maternal history of breast cancer. Her mother, aunt, and many more has died out of that. So she has done this thing so that she did. I'm talking about like 10 much many. Today, genome sequencing is very very frequent, but in those days, like say 7 8 years ago, it was not very frequent. So she spoken to a particular lab to had her BRCA gene sequence, and she found out that there is a particular mutation, and this mutation may be fatal, and therefore she has got rid of her tissue completely. And since she is an actress, she is now using a prosthesis. So we'll come to once again to this thing, our discussion. So this is the BRCA gene. So we are having the ability to use artificial intelligence to predict whether a particular, and one more important aspect is not all mutations are cancer. So it could be a mutation, but it could be a silent mutation. So what exactly happens is, in by genomic sequencing, you are identifying that there is something wrong in the sequence of the gene, but it may not be fatal. It may be there that this is a silent mutation and there is no effect on the protein. If there is no effect on the protein, there is no significance of this particular mutation. So how exactly you will decide that which mutation is good and which mutation is bad? Understood. So in that case also, we'll use a significant amount of machine learning tool and technique to predict how exactly a particular mutation is good or bad. So in that particular what we call domain of diagnosing. Okay. Oh, okay. So I'll come back. So what exactly happens is, just give me a minute. Okay. So so in this particular domain of predicting the SNP, whether it is cancerous or not, this particular university, Harvard, the genetics department has done quite a tremendous development by deploying machine learning tools to predict the outcome of a particular mutation. So we'll share with you a video. I thought of doing a hands-on, but due to some limitations, I could not. So, madam, may I request you to please play the next video or the simulation? I think there is a simulation. So, madam, please click on the link and share your
Sure, sir.
screen so that you can see the gene how it looks like. You can even share the link to the participants in the chat box. If they are interested to see, they can also able to see it, that how the genomics of genomic data of BRCA looks like. Yeah,
Sir, is
the
this
simulation one I think I have given. Yes, you can directly click on it. Okay, now we are entering into a database. As I have said, this is a real-time database which is curated continuously and it is updated regularly. Now, this is the reference genome of BRCA. So, breast cancer antigen, this is the reference sequence. Reference sequence means what? Without any mutation. Okay. So, this is called a particular, like, this is called the GenPep format, where they have written all multiple information, where it was sequenced, what is the technology that was used for the sequencing. For example, let me tell you the publication status, and then many other things, whether some patent where they are, which kind of patient was that a clinical trial or not, multiple different information. I'm sure many of you are already aware of. So this is so many information in this. So in the right side, if you can see, there is the structure of the protein also, isn't it? Okay, it
here it
was
is?
there, I think. Okay, so there is the structure of the protein also, 3D X-ray crystallographic structure. Yeah. So this is also there. So you can use these tools in a way you want it. So now we'll make it into a more usable form. As you can see here, there are 90 amino acids in the whole protein. So you can think the severity. Only 90 protein, 90 amino acids. If a single amino acid alters, there will be a huge lot of problems that may be associated with that. So what we'll do is we will use the FASTA format. Madam, please click on the FASTA so that all unwanted information will get reduced. So we call it like a data normalization because we only want the sequence. So when you click on the FASTA, it will just give you a simple the simple single amino acid names, like it started from L, leucine, tyrosine, phenylalanine, aspartic acid. Like this, it is going on. So there are 90 amino acids. If one or two amino acids is mutated with something else, there will be a huge loss in the structure of the protein, and eventually it will lead to breast cancer, which has been the case in the family of Angelina Jolie. So now we'll go to one more video, Madam, video number 3, where we'll try to show you how exactly the Harvard genetics team is using machine learning for this particular prediction whether a particular mutation is good or bad. Okay, now this has a different aspect. Madam, I'll request you to put it a little this thing fast forward कर दीजिए madam. Um,
फास्ट
so
फॉरवर्ड
these are different issues. हाँ मैडम फास्ट फॉरवर्ड कर दीजिए, थोड़ा आगे लेके आ जाइए so that okay,
ओके सर
let's see what is there. Yes, okay, these are different proteins. This video has been produced by our lab only for a particular kind of requirement. So these are some structural aspects of the protein where it interacts with a particular ligand, and we can also able to find out whether the ligand binding is precise or not. Madam, a little bit more.
श्योर सर
Fast forward, madam. Okay, these are how the ligands are interacting with the particular protein. So it was a different aspect of drug discovery. But since I'm not discussing it, we will not discuss. Okay, from now here you can play. अभी थोड़ा आगे जाके a little bit more where we are seeing the gene sequence. Fine, madam. Now you can play it. Okay, so this is of a different aspect, but here again it is also the sequence of a particular protein. Now this is the tool. Saran madam, please see, participants. This particular tool has been developed by what we call as Harvard Genetics Lab, which uses a particular kind of technology which is called naive Bayes algorithm. Okay, so what exactly it is doing? It is trying to find out whether a particular kind of mutation in a gene will be bad or good. Okay, so what exactly they are trying is, it's a very simple tool which runs on the concept of artificial intelligence. The specific technology is known as naive Bayes algorithm, or we call it a probabilistic classifier tool. So what exactly they have done is, they have made a window where you will simply paste the sequence of the protein. Okay, just hold on. Madam, am I audible?
यस सर यस यू आर
Okay, fine. Now, in that tool, you can simulate the mutation. Okay, so I'll once again tell you what exactly is going on there. So this is the sequence of the protein. It could be any protein like BRCA. Okay, so in this example, it's a different protein. So we can use BRCA. Now we are doing the simulation. Madam, just 1 minute ये कीजिए, previous दीजिए madam, just 1 minute previous.
इज इट इज फाइन
Madam,
सर
in a little previous madam, in the first, just pause it for a 1 second so that I can, madam, थोड़ा आगे,
ओके सर
uh, previous madam, previous window.
इज इट फाइन सर
Madam, please pause for a minute. Pause for a
हाँ
minute.
आई ऑलरेडी
Okay,
पॉज
okay. So what exactly? Sufficient. So what you can do here is, the tool is so simple, and this is done by the funding from that lady, as I have spoken about. So what exactly happens is, in this particular toolbox, we can simulate the particular kind of mutation. So how exactly we'll do this? Please see. So there are position. Position means at which amino acid you want to do the mutation. Okay, it's entirely up to you. So in this particular example, we are doing a mutation on the 10th amino acid. Okay, please see. So on the 10th amino acid, what was the previous one in the non-mutated? That is in the novel condition. What was the amino acid? D. Okay, see, there is a substitution pain. Can you see? In the substitution, there is AA1 and AA2. In AA1, we have selected D because on the 10th amino acid, it is aspartic acid. Aspartic acid is represented as the digit D. Now, what you'll do is we'll change the aspartic acid into R. R is arginine. Okay, so aspartic acid is a highly negatively charged amino acid, and arginine is a highly positively charged amino acid. Okay, so for the doing of the experiment only, we are doing it. It is not the case of a patient. So what we have done, we have replaced a positively charged, a negatively charged amino acid with a positively charged amino acid in a simulated what we call experiment. And now, when we will click on the run button, that is the calculation will start. Now here there is a beauty of this particular tool. Why I am discussing it. So this particular technology or artificial intelligence tool uses a method which is called as I have said, probabilistic classifier method. Now, in that probabilistic classifier method, what exactly happens is this is a software which runs on a web server. Okay, so you need not to download a whole software into your system and then use your computational tools. So what usually happens is there are two kinds of software. One software which is present on the internet only through a web server. For example, your networking to say, like for example in your social networking uh, social networking tool or social networking sites, you do not need to download everything. Only a small this thing, what you call application, you download, and eventually all your task is done. So it is something like that. You need to download the whole software, which will use a lot of space, lot of computational tool. Instead of that, you use a web server. Now, in that server, what you can do is you can simulate a whole process of identifying a particular mutation, whether it is good or bad. Now, how exactly it is doing it? Because whenever you are using this particular what you call software, you are using the grid computer or supercomputer of Harvard University. Okay. Now, madam, please play the video. So what we are trying to say is here the simulation is done by using a particular kind of mutation where we are replacing D with R, and we want to see what is the result of it. So it has entered into the grid computational network of Harvard. That is Harvard supercomputer system. And then it will give you a result. Okay. So this result will be like this. The result is what? As you can see here, the position 10. On the position 10, you have replaced D with R, and this is the result. It will be probably damaging. Okay. And it has given you a score also. Can you see the score? So what exactly this particular tool is giving you? It is giving you a ability to predict whether a particular cancer will be particular mutation will be good or bad, whether it will lead to a cancer or not. Understood? And by using what? By using a machine learning application. So thank you, madam, for sharing it. I'll now go to the my presentations pane once again.
Sure, sir.
So I'm going to the discussion once again. Just one second, madam. I hope that our participants have. Okay, so this is the tool that we can this thing. So what is the overall objective? The overall objective is to make more better predictions. So there are different algorithms that are being used. So this is the exact algorithm which this particular tool is using. This is called probabilistic classifier. Now please understand how machine learning works. Machine learning works with three different things. One, a statistical likelihood or probability, then the prior experiences, okay, and then the evidence that is there with you currently. So machine learning is not much different from human learning. Human learning is also like this. If you have some prior experience, from that experience only you behave. This is exactly the same. So here what exactly it is doing is, it is identifying a case or predicting something on the basis of the likelihood, and then the prior experience it has, and then the evidence, the current evidence that we are having. On the basis of all this, by using a training set, this particular algorithm tries to understand whether a breast cancer mutation will be really a cancer, or it will be a silent cancer, a silent mutation where there will be no problem at all. Are you getting? So this is a very good example how machine learning is getting used or being used for diagnostics purpose. So this is a particular naive Bayes algorithm that I have said. Here also there are several benefits as well as some limitations. Because the limitations are not that much, benefits are more. That's why we are using it. And in combination with something else, there will be excellent correctness of your prediction. Okay. So in 2013-15 also, we were given a task to find out some real problems that was been there with a particular heart disease, which is called birth syndrome, which is a genetic disease of abnormality in heart. And we were being given a particular some amount of fund to understand what exactly has gone wrong. And eventually we found out the reason, which was nothing but a mutation in a gene called tafazin, which is nothing but a protein present on the outer membrane of mitochondria. So we eventually reported it, and eventually there has been very good response of our work. Okay. So this is again one more concept which is called this is again support vector machine, where we can differentiate and classify between two different occurrences. Here there has been given an example again of cancer only, to predict whether it will be a malignant or benign. And this is a much more discussion of this support vector machine, which is an algorithm of machine learning environment. Mm, so this we can use for multiple, not only for cancer research, also to identify whether a vaccine will be good or bad. So now I'm giving you the second example. As I have said, I'll be discussing two things. One, the diagnostics of cancer, and then how very good vaccines can be developed against this COVID. So why, if we see the mechanism how COVID develops, we can also use machine learning for predicting a very good vaccine against it. Also, these are in rudimentary stages, but let me tell you, previously when usual time taken to develop a vaccine is approximately 10 years, because there are different phase trials will be there, then there will be multiple different things. But this year, as you can see, in 1 year only, the anti-COVID vaccines were there, isn't it? So how this was possible? This was possible because there was very good artificial intelligence tools and techniques which have helped us to identify which will be a stable epitope to design a particular vaccine, which with sustained what we call is immune response. Okay, and to sustain production of memory cells, which will eventually help you to get protected from a particular disease, a particular infection. So how exactly it is happening? Please see that the usual process of identifying a vaccine epitope, there are some repetitive tasks. Why? Because the exterior envelope of the virus has different spike proteins. Out of the many spike proteins, a particular spike protein only will elicit much more immune response. So not all the spike proteins that are there, isn't it? So usually how exactly the binding takes place with a particular spike protein, a receptor called ACE2 receptor, okay, ACE2 receptor will bind, and eventually there will be an internalization of the virus into your lungs lining, and then inside your lungs so many viruses will be replicated, isn't it? So what will happen? Your body will have a huge burden of COVID virus, and eventually you will show multiple different signs and symptoms: reduction of oxygen concentration in your body, dependence on exterior oxygen, and so many other things. Eventually the patient may also die. So what is the goal of artificial intelligence? To find out a particular stable epitope on the surface of the virus, which will elicit very good immune response against this particular virus. Okay, just hold on. So in the next this thing, there is okay. In 2020, we reported our observation on 19, which has been also showcased by World Health Organization, and we are very much indebted to it that they have considered our work significant. And then this is the NCBI virus tool where we are having the genome sequence of Wuhan virus. Okay, as you can see, this is the severe acute respiratory syndrome coronavirus 2 isolate 1 complete genome. The complete genome sequence is here for this particular virus. Okay, so we can see it that again it's a huge this thing. I am not sharing it right away. So there are now this is very important. So severe acute respiratory syndrome coronavirus 2 isolate SARS-CoV-2. This is the surface glycoprotein gene, particular surface glycoprotein gene, which the sequence is there, and which will eventually give you a very stable epitope to make a vaccine out of it. All the vaccines, that is subunit vaccines, Covaxin, Covishield, both are made on this particular what you call protein sequence. Okay, so we'll also come to this particular point, and then we'll see how exactly the whole process has been taken place. So, madam, once again, may I request you to please share your screen and play the next video. Madam, also in the meantime, please tell me how much time I have been given.
Further 26 minutes is there, sir.
How much?
26 minutes, sir.
26. Okay. I'll cover it up. Madam, please play the next video. It will
Sure.
give us a very small this thing that how exactly the infection takes place. So why this is important? Because if we know the pathway how exactly infection takes place, we can also develop very good vaccine out. Okay. Skip. Okay. So this is okay. So no, madam, this is an I think not the video. Okay, okay, right. Yes, this one only. Yes, madam, this one only. So this is the virus, as you know, and on the surface you can see multiple different spike proteins, isn't it? So these spike proteins will bind with this ACE2 receptor. So what is the role of this small molecules that we are looking for? It will stop, or it will interfere with this particular binding. For example, initially they have used chloroquine phosphate. If you can remember, there was a case where America was asking more and more chloroquine because India produces huge amount of chloroquine, and it was been seen that chloroquine interferes between this particular binding. In the video, you can see ACE2 receptor is binded with one of the glycoprotein of the virus. So this particular, if you can interfere in between this interaction, what will happen? You can protect yourself from this virus. However, it was not very significant and not very useful at that point of time. So what exactly happening was this interference was not very good, isn't it? It was been binding with the ACE2 receptor, chloroquine phosphate, but it was not that good that it can interfere. So all on a sudden, initially chloroquine phosphate was given as a standard treatment, but later on it was been stopped. I think all of you already know. Then it came ivermectin, if you can remember, isn't it? So ivermectin was given again because it was having some interference with ACE2 receptor, ACE2 receptor. So eventually it also failed, and therefore it has also been removed. Then lately it came remdesivir. Now remdesivir inhibited the multiplication of virus. So in that case, it was not interfering with the binding of the receptor, but with the replication of the viral DNA inside the host. Okay, so remdesivir is nothing but a nucleotide analog, which is analogous to guanosine G of the DNA strand, and remdesivir is nothing but a structural analog of guanosine, and therefore it was trying to inhibit the viral replication, and therefore it is still now the standard procedure to treat remdesivir by remdesivir. in COVID patients. But rest of the other two, which interfered with H2 receptor binding, was been removed from the protocol of ICMR and international WHO also. So, madam, I'll once again request you to play the next video or the simulation. I think there is a simulation. What was there, madam? Please, would you play?
I have simulation too, sir. Is it,
Please,
sir?
madam, please click on this. Yes, madam. So that is nothing but the COVID-19 genome. That is the first genome. You can remember without any mutation, no alpha, no beta, no omicron, nothing. The pure COVID-19. Okay, we against which we have all taken 2 jabs of vaccine. Okay, I think you are already sharing.
Yes, sir.
Okay. Madam, have you shared the screen?
Uh, Wait a second. You getting that, sir?
Okay, right. Yes, it will come. It has not yet come. Okay. Okay, madam, shuru se jaiye. Okay. Yeah, this is the whole genome sequence of this thing. COVID-19, as you can see, severe acute respiratory syndrome coronavirus 2. If you can work with this and play with this, you can definitely use it. Madam can also share the link of the simulation to the participant. And you can see this is again the NCBI database where the whole genome sequence of this particular virus is there. Not only the whole genome sequence, the proteome is also there. All different glycoproteins of the surface, which has been sequenced and identified, is also the data is also there integrated. Okay, so you can use it if you are liking to do something with this. So we are not going into much detail. So from this genome sequence only, we finally used the sequences to design 2 vaccines, Covishield, Covaxin, which were nothing but what we call is subunit vaccine. So, madam, I'll request you to please play the next video.
So can I play the next video?
Yes, madam, please, madam. Yes. Uh, so what exactly happens is a protein or the this thing what we call is this particular epitope, something looks like this, which is having different areas, binding pockets, this and that. Okay, but the problem here is, what is the problem? Problem here is, when you are designing a vaccine, it should be very stable. The stability of the vaccine is very important, let me tell you. So I'll give you one more example. Most of the vaccines should be preserved under a particular kind of temperature. Why? Because the protein is highly thermos what you call sensitive, heat sensitive. If you are putting so many stabilizers, buffers in the solution, still it can go denatured. Therefore it is having certain shelf life. Okay, it is having certain shelf life. So how will you think, for example, you have isolated a what do you call a stable epitope, and then you are going for the vaccine development. But in the middle, you see the protein is so fragile that it is getting distorted and there is no stability in the protein itself. So what is the use? What is the use of this vaccine? The vaccine was very good to elicit immune response and to make you immune from the disease, but it was not stable, and therefore you cannot transport the vaccine from one part to the another, which is a major drawback. So what you need to do is you need to finalize which particular epitope is very stable. So out of multiple different, these are small parts of the epitopes that we have identified on the surface during the WHO study. As I have said, in 2020, we did a study on this epitope and how it binds with the HLA, or human leukocyte antigen, or MHC1 molecule of human. We studied that. So from that only, one video has been prepared for our students and other things. That video we are demonstrating. Okay. So these are 2 epitopes taken. Okay, both the epitopes will look like same, as you can see. Okay, both. See. So previously this was the skeleton, and then now this is the what we call is tertiary structure. Okay, we are using a machine learning tool which is very simple to use. So what exactly happens is, we have already instructed the tool that is the software, to identify the delta G value of each and every atom. Madam, please give a pause for 1 minute.
Sure,
So what exactly happens is, now what exactly happened is, as you can see, these are both epitopes of the same virus, but one is having very high stability and another is having very low stability. So how you will understand which is more stable and which is not? According to laws of thermodynamics, it says that if in any atom the degree of disorder is reduced to less than zero, the molecule will be more stable. In the contrary, we can also say if the delta G value or the free energy value of any atom is less than zero, it will be more stable. So according to that notion, this particular toolbox, okay, this is nothing but a machine learning toolbox which you can teach. So we have taught it to understand or to color different atoms in different color if the delta G value is certain. So from the X-ray crystallography data of the epitope, we have found out the delta G value of each and every atom. And then we instructed the tool to color it different colors. So in the right side, you can see there is completely blue structure. Now this blue structure is blue because we have instructed the tool that if at all you get more than zero delta G, you make it blue directly. Thumb rule. What is the thumb rule? Delta G more than zero is definite. That means positive in magnitude is blue. So in the right side, you are having an epitope which is very unstable. But on the left side, you are having an epitope which is a different color. Can you see? Multiple different colors. All the colors of rainbow is there. Why? Because each and every atom is having different delta G value or free energy value there, which it indicates that the whole protein is comparatively more stable than the right side protein. Okay, the blue protein is less stable, but the multicolor protein is more stable. Why? Because it is having a in most of the atoms the delta G value is negative. So how do we understand that? Now this is a static. What do you call? You cannot really see how the protein will behave. Understood? So there is one more technique in computational prediction or biomolecular simulation, what we call as molecular dynamics. In molecular dynamics, what we do is, now here the protein is stable. Can you see? We are doing the experiment in vacuum. Okay? So the whole experiment that we did in this video in vacuum, but we really need to understand that in real conditions, that means where the protein is suspended in blood, inside the blood, how it will behave, inside the buffer, how it will behave. That means not in vacuum. In vacuum, anything can happen. But in real time, in real conditions, how the behavior of the protein, that also should be understood. So that we will do by a method called molecular dynamics, which is again a computational tool. And in 2013, the Nobel Prize for Medicine was given to this particular tool. So we'll come to this thing again. So, madam, please play the video. I think it will end soon. हाँ प्ले कर दीजिए मैडम.
sure, sir.
Oh, I think okay, fine. So this was a static simulation, okay, of the what you call is surface epitopes or surface glycoproteins of COVID-19. So now, madam, we'll go to the next video. Okay, this is fine, madam. This is the whole sequence, and from that a small particular simulation was done. This video is also generated by our lab. Okay, now skip कर दीजिए मैडम, skip कर. Okay, now this is called molecular dynamics. In this, what we do? We are no longer putting the protein in vacuum. We are putting it at a solvent. We call it a solvent box. Inside that solvent, we are putting the protein. So as you all know, please this thing that in the solvent there will be plenty of different forces of nature that we call. There will be van der Waals interaction, there will be ionic interaction, there will be hydrophobic, hydrophilic, so many more. So due to that stress, the protein will show you the real behavior of the protein. Not only computation, it is a real story. So in that story, what you are seeing that the whole structure of the protein has been broken down into small fragments. Can you see all the small fragments? Is the strongest part of the protein that is still remaining. It's a 15 picosecond simulation that we did and then made screenshots and put it as a video by a particular software. Anyway, so in that video, what is showing? It's showing that for 15 picoseconds, we put the protein in a solvent, it will be the behavior. All thing will be destroyed. There will be nothing as the vaccine. So this is a very good candidate. So we'll go to the next video. Madam, next video link, please share. No, madam, this is you close.
Can I play the next
Uh,
video, sir?
you close this, madam, and next. Yes, right. Yes. So now this is again the protein with higher stability. So what is the behavior? Here you see. It is also getting destroyed. There is no denying. But you can still see the secondary what you call loops. Can you see the alpha helix? We call it, or we call it this thing. Yeah, alpha helix. Can you see? This is the secondary structure of the protein. That means it is more stable than the previous one, and therefore, instead of the presence of different forces of nature, the protein is still trying to maintain its integrity. Are you getting? So what exactly is happening? We are having to we are seeing it that there is a certain more domains which are more stable. So on the basis of all these things, what you can do is we can mm, uh, predict which protein is better for our what do you, thank you, madam. Now I can go to the last 2 3 slides. Thank you, madam, for your excellent coordination. Okay, okay, okay. So that was the thing we tried to explain, means try to show you now how that software works. It combines multiple different tools and eventually gives us a pattern according to our instruction on. So we call it pattern recognition. So these are multiple different things. Okay, now this is the thing that we spoke to you, one stable and one unstable. And one more important aspect, I'll just do it in a very fastly. So this is called decision tree algorithm. So decision tree algorithm, how it works, is a very important algorithm. So see, this is the case for cancer diagnostics only, with a very simple demonstration of the concept. So the lump, the patient will be told to do self-examination. If there is lump, so there are two options. There is no lump and there is lump. So if there is a lump, the patient should go for a mammogram, which is a scanning of the tissue. If there is no lump detected, then self-examination should be done after 1 month. Then if the answer is yes, the person should go for a mammogram. Now, mammogram can be suspicious or there will be normal study. If it is normal study, no biopsy is needed. But if something is fishy or not within the normal range, then the patient should go for a biopsy. Okay. Now, if the on the basis of the biopsy, it could be two. Either that could be a non-malignant cyst, which will be reduced after medicine, and it could also be a malignancy where it should get rid of the whole tissue, or there will be a major operation. Okay, so this is how a decision tree algorithm works. But the problem here is we have to use multiple different tools, concepts to finally simulate a whole biomedical research pipeline. So in order to integrate all, we on 2020-21 have written a patent and we submitted and we are um, about to get it granted. So this is the whole architecture of the method where we can integrate multiple different aspects, and so that different algorithms need not to be used. So we have already submitted it and we are talking to these small scale industries to monetize our product and to do it for wide circulation so that we can build a business out of it. So these government registered MSMEs have also spoken to us, and we are in discussion to what do you call to make it a monetized thing, and we have also had a round of shall we say initial discussion with IIT Bombay for a innovation challenge, and the challenge is still going on. So these are some of the references that we used for our previous studies and all. And these are the things that I wanted to share with this August gathering of participants. And I'm thankful for the coordinators for giving me the opportunity. And I'll be happy if there are some questions from the participants, and I'll try to answer them if there are some. So by saying so, I'll complete my discussion, and it's over to you, the coordinators.
Thank you, sir. Thank you so much for your excellent session. I request the participants.
Excuse me, ma'am. Devna sir, can you please switch on your video?
Hello, Devnath sir, are you there?
Yes,
Dev,
yes, madam. Boliye.
sir, please switch on your video, sir.
Okay, madam. Due to a little this thing, I'll request you not to this thing now.
Okay, okay,
But
not an issue,
on
sir.
to if there are questions from participants,
Yes, sir.
I hope I do not know whether this was the discussion was relevant, but I could not resist the idea to discuss these developments with our this thing, and I congratulate sir on to
No, no, sir. It is
conceive
very
this particular
it is very
terminology.
relevant to our title, sir. And the session is very
Okay,
excellent,
okay.
sir, and we have got a very good feedback. And I request all the participants, if you are having any queries, speaker sir is there to clear your
Yes,
queries.
please. They have some query. And
Yeah.
one more aspect of this outreach activity is to collaborate with people who are having similar kind of approach or some development in that field. So that would also be great if somebody is there who can would like to have some discussion with us. We can also able to forward our this thing. Madam, most of the participants are from pharmacy background, madam.
Yes, sir. Yes, sir. Most of the participants are faculty, sir, from the pharmacy department.
Yes, yes, I understand that.
Yes, sir. Yes, sir.
This is for faculties only.
Yeah, no, not
For
only
the
faculty, sir,
participants
for
who
students,
still
but
remain
70% are faculty members, sir.
right. Okay.
Uh, Thank you, sir. Thank you for your excellent session. We are
Okay.
very, from our management and from our principal coordinator and co-coordinator and from our staff, we are very thankful to you for giving this wonderful session, sir. And I
Yes.
request all the
Thank you.
participants that session one feedback and assignment is posted in the chat box. And be here. The next session will start within 5 minutes. That is at 11 o'clock. I
Acha,
request
madam, I
once
had
again.
sent you the MCQs also. Have you received?
Yes, sir. Yes, sir.
Okay. Okay. Thank you, madam. Thanks a lot.
Sir,
Okay.
just a minute. Just a minute, sir.
Yes.
Sir, can you see the screen, sir?
Yes. Yes. It has come.
Slideshow.
Okay.
Yes, sir.
Okay. Thank you,
Uh,
madam.
Thank you so much, sir. Thank you so much for delivering this piece, sir.
Thank,
I repeat
thank
once
you, madam.
again. The participants, the feedback and the today's attendance is posted in the chat box, and the next session will start at 11 a.m. Thank you. Thank you so much, sir.
The next section will be started at 11:15 a.m. Kindly attend the participants on 11:15 a.m. for second section.